Welcome, everyone, again. Thank you for joining us today. My name is Leo Alfonso. I work in the International Relations and Partnerships team for the STEM disciplines here at ANU. We have my colleague Chami on the call on the line, and of course, we are joined also by Professor Thomas Preiss. I'll do a proper introduction in a minute. Before we get started, I'd like to take a moment to acknowledge the Ngunnawal and Ngambri people who are the traditional owners of the land on which ANU is located. I pay my respects to their elders past, present, and emerging. All right, so Professor Thomas Preiss, we're very lucky to have him with us here today on this call to share his time and his knowledge with us. I'm sure most of you have already read his bio, so I'll keep the introduction short. Professor Preiss is a world-renowned expert in RNA biology. He completed his PhD at the University of Newcastle in the UK, followed by postdoctoral work at the European Molecular Biology Laboratories in Germany. Before joining ANU, Professor Preiss was laboratory head at the Victor Cheng Cardiac Research Institute in Sydney. In 2011, he joined ANU as professor of RNA biology, and since 2022, he has been the inaugural director of the Shine Dalgarno Center for RNA Innovation. Today, he will explore the science behind mRNA vaccines, and he will offer some valuable insights into this field of study. And with that, Professor Preiss, I would like to hand over to you. Thank you very much again. I'll stop sharing and let you take over. Thank you.
All right. Thank you very much, Leonardo, for the introduction and for having me for this webinar. And I thank everyone who signed in for it and agreed to spend their time with me for the next 50 minutes or so. I will now share my screen. I hope this is working.
It's working. I stay online just to give the thumbs up and then switch off camera.
So, yeah, so I prepared a series of slides to kind of bring everyone into the science behind mRNA vaccines. And like I already mentioned, I'm an RNA biologist working at the John Curtin School of Medical Research here at ANU. And for my presentation, I've structured it into 7 or 8 kind of sections. I will give a very brief overview of COVID-19, the virus that caused the recent pandemic. I'll run you through some vaccine types that have been developed to vaccinate people during the pandemic before focusing on the mRNA vaccines and how they work. And to help you appreciate a little bit more how mRNA vaccines work, I have a section on our genome, our DNA, and how mRNA is a messenger or a transient photocopy of the blueprint of the genomic information that we all have in our genome. I'll run you through how mRNAs function as templates for protein synthesis, and then I'll explain a little bit more the specific features of eukaryotic mRNA that need to be replicated in an mRNA vaccine for it to be successful, before finishing off with an outlook on what else is now possible with mRNA therapy. And if I have some time left at the end, I also have some bonus slides on some other RNA discoveries and achievements that were made here in Australia. So, okay, part 1. So COVID-19 is actually an RNA virus itself. So what you can see here in the middle of the slide is a schematic of the viral particle with the RNA genome inside of the particle and some other key features and proteins, particularly the spike protein, which is what the virus uses to dock onto our cells and gain entry. And so consequently, the spike protein has also been the feature that was particularly selected to generate an immune response to with vaccination. But maybe as an interesting aside here, while much of, well, all extant life forms run on DNA as the genetic material, viruses are likely a kind of a remnant of an RNA world where RNA itself was also used as a storage molecule for genetic information. And they are still storing their genetic information in the form of RNA. Not all viruses do, but the RNA viruses do that. So what are the types of vaccines that have been developed to try and deal with the pandemic when it emerged? Now, it followed by and large an established pattern or an established set of technologies that are available to make vaccines. And basically all of these options were tried and led to varying degrees of success and use in different countries around the world. So the basic idea with a vaccine is, of course, to introduce some sort of harmless parts of the pathogen that you want to immunize against into the body so that the immune system of the body can develop a response and a memory of that response prior to an actual infection. And then when you actually do get infected with the pathogen, you can battle the pathogen rapidly so that either you're not getting sick at all, or at least the course of the sickness is much milder than it would be without prior vaccination. The most classic form of vaccination is in the form of an inactivated pathogen or virus in this case. So what was actually done successfully in several countries around the world, like China, Russia, and Iran, is that scientists and biotechnology facilities managed to grow the COVID-19 virus in bioreactors. And then they purified the virus from the cell cultures, and they killed the virus, in quotation marks, using heat or chemicals. So basically that is an inactivation of the infectious potential of the virus, but leaving the key features of the virus intact so that the immune system can recognize it as foreign. So what this type of virus really is an inactivated form of the pathogen that is then introduced into the body to create an immune response. Now, another type that was used early on in Australia, particularly as well, is what became known as the Oxford or AstraZeneca vaccine. So this is a viral vector vaccine. And how that works is that a protein that is typical or a recognizable feature of the pathogen, in this case, the COVID-19 virus, is identified as a suitable antigen. Then a DNA sequence is decided on and manufactured to represent coding potential for this protein. And this DNA sequence is introduced into an unrelated harmless vector virus, adenovirus in this case. And then these harmless viruses that carry now a, let's say, transgenic payload of the protein encoding the protein you want to vaccinate against. And you can immunize people with this viral vector vaccine. The harmless virus enters the cell in the body, and the cell produces a certain amount of the spike protein, and then the immune system of the body can develop an immune response and a memory of that to it. Now, another version of a vaccine that was also successfully developed and then deployed is the so-called protein subunit virus. So in this case, again, the spike protein was identified as the suitable antigen, and that spike protein was produced as a recombinant protein in a bioreactor. And that protein was purified, formulated with an adjuvant to boost the immune response, and then that is injected into patients as a vaccine. So here you have, in a way, the most direct representation of the antigen in a form of a vaccine, because the protein is made before the vaccine is introduced into the patient. So most direct, other than using an inactivated virus. And finally, and what has become particularly versatile and successful, as it turned out, there are the mRNA vaccines. So again, you start off by identifying a protein that is going to be a suitable antigen. You generate, in a series of steps in the laboratory, a messenger RNA that encodes for this protein. You formulate that messenger RNA into a vaccine, and you inject that into the patient. So here the mRNA enters the cell, and the cells read the instructions on the mRNA to make the protein that is then recognized as an antigen, and the immune system generates an immune response to that. So this is just like a brief overview of different vaccine types that have actually been developed and deployed during the pandemic for COVID-19. So how is an mRNA vaccine formulated to be a viable vaccine specifically? Well, a lot of attention has to be paid on to how exactly the mRNA is engineered as a template for cellular protein synthesis by ribosomes. And I'll explain how that works in cells a little bit later in my talk. But basically, the mRNA has to have a main feature, which is the payload or the reading frame, the sequence that encodes for the protein you want the cells to produce ultimately. And then it has to add, you have to add various other features that are commonly found on cellular messenger RNA, so that the cell will know what to do with this messenger RNA once it gets introduced. And now there are many different further developments improving on the, let's say, first generation mRNA vaccines that were developed and then used. So all of these features that are listed here as bullet points can be somehow tweaked, perhaps to give a better mRNA vaccine, a more stable mRNA, or an mRNA that is translated better. And then there are some other features or versions of mRNA therapy that are now also being developed. There is something called self-amplifying mRNAs that combines a little bit of viral biology with the mRNA principle to basically introduce an mRNA into cells that can make copies of itself and therefore generate a higher yield of the antigen protein in cells, as you would normally expect from that level of dose of mRNA. And another thing that is quite interesting is that if you make the messenger RNA into a circular molecule, that could have certain advantages in terms particularly of the stability of the messenger RNA once it gets into the body. The other feature that is quite tricky, and again also has much innovation potential still, is how the mRNA is packaged so that it is, first of all, somewhat stable on the shelf, but then also is stable in the extracellular fluid when it is injected as a vaccine formulation, and then also so basically an mRNA in this current version of the technology is packaged into a lipid nanoparticle, as we call it, which stabilizes it on the shelf. It also helps with stability in the extracellular body fluid, but also helps with entry into cells. And so here you have a kind of a first schematic of an LNP particle, and I'll explain that a little bit more in a later slide. But basically, it is like a little fat bubble that contains the mRNA inside. And then when this fat bubble fuses with the cell membrane, it can release the messenger RNA into the intracellular space. So why is mRNA actually a suitable vehicle to express an antigen in cells? That's because it is a transient copy of the genetic information that we carry in our genome. And so now I have prepared a set of slides to try and explain how, in principle, the genome functions in cells and how the messenger RNA comes into that picture. So what you probably all know is that our genome is key to the heredity. The same genome is present in nearly all cells in our body, but it has a special role in the germ cells. So it enables the propagation and the conservation of a species. And at the same time, with sexual reproduction, it also enables recombination between paternal and maternal genome to enable genetic diversity in the population. A mutation of the genome in germ cells in particular, it has also a very useful function in evolution, in that a slow rate of mutation over evolutionary time enables adaptation, but also ultimately the evolution of new species. But of course, in the more human timescales that we exist, mutation in germ cells and the propagation of this modification is also the cause of genetic disease that runs in families. But what else is the genome up to the rest of the time when it is not about generating the next generation of a species? Well, it is actually the informational molecule that carries the instructions for the running of all the cells in our body, right? So you have here kind of a circle that I made up with human bodies here at the bottom left. We consist of an incredibly large number of cells, many different cell types. Inside the cell, in the so-called cell nucleus, you have the DNA that has all this, stores all this genetic information. But the cell consists of many other compartments and components, not just the DNA, but also RNA and protein, lipids, and so on. So all of that is generated based on the genetic instructions in the DNA. The DNA itself is not one large molecule for the entire genome, but it consists of multiple chromosomes. So each chromosome is a continuous double helix of DNA packaged up with histones and compacted into a particular shape. Humans have 23 chromosome pairs. In this large amount of DNA, there are about 20,000 or so genes that encode for proteins, which are the main workers in the cell, as I come to next. So a gene, as this historically has been defined, is a section of DNA that contains building blocks of the genetic code, the 4-letter code of DNA, GATC, the 4 letters. And these genes are instructions to produce protein, which are the key workers and the key drivers of building cells and building an organism from many cells that have specialized roles as well. So what's a gene then in our genome? I use this kind of well-established analogy here of a book. So if you think about our genome as an entire library of many books, then you could think of the individual chromosomes in our genome as sections or shelves in that library. A book is maybe a gene, and the genes are also split, as I will show you in a subsequent slide. And so chapters in that book could be exons. And if you had some blank pages inside of that book between chapters, that could represent what we call the introns. And the letters that constitute the text in these various chapters, these would be the bases, A, G, T, and C in various sequences. So how is that genetic information in the genome actually put to use in a living cell? Now, the principal way in which this is done is that there are enzymes called RNA polymerases that can transcribe sections of DNA or genes into a less stable copy of those genes, what we call messenger RNA. And these messenger RNAs, they leave the nucleus where the DNA resides in the cell and go to the cytoplasm where they meet the ribosomes and associated factors. And ribosomes are these little nanofactories in the cell that can read the code in the messenger RNA and can make protein. And proteins are, they have many functions in cells. They are enzymes catalyzing chemical reactions, but they also carry out like mechanical work in the case of muscle cells and many other processes that are a feature of life driven by proteins. So one problem then comes up, which is in a highly specialized multicellular organism such as us, we have many different cells that carry out specialized roles in our body. We have different tissues, and some of these tissues consist of multiple specialized cell types as well. So I think on one of my earlier slides, it said that we have about 210 different cell types in our body. So how can it be that the same book or the same collection of genes that is present in virtually all cells in the body in the same way can instruct different types of cells to carry out very specialized roles, to have very specialized phenotypes, as we say? Now, the analogy that I came up with here is that if a genome was a book, like a large cookbook that contains all the possible recipes of all the dishes that might be served around the world, then there's a process during embryonic development where an epigenetic process, a chemical marking of sections of the genome will close off sections of your large cookbook library to end up with cells that can only make dishes of a particular variety, which is suited to that function of that cell type. So in this analogy, I don't know how well it works, but basically, as a cell differentiates during embryonic development into a specific cell type, it goes from being able to cook any dishes around the world to just one particular ethnic cuisine. Let's say a particular cell type can only make Chinese food, and another cell type can only make Italian food, and so on. So that's how during development cells take on specialized functions in the body. And the menu of a restaurant in this analogy will list all the possible dishes, all the Italian dishes you can order in an Italian restaurant would be available on the menu in that cell type, if you like. But then in addition to having that sort of permanent specialization on a particular cuisine or cell phenotype, you also have to react on a day-to-day or minute-to-minute level to changes in the conditions that the cells experience. So there has to be a dynamic change of reading of recipes or gene expression in response to conditions. So in the restaurant analogy, every night you have a different set of customers coming in the restaurant, and they might order a different subset of dishes in different numbers. And you have to tell the chef in the kitchen which dishes to cook and how many. So that's how a messenger RNA functions, really. It is, in this analogy, a photocopy of a gene in the cookbook, in the DNA, that is made to order. Basically, if the cell demands the production of certain protein, the RNA polymerase will make a certain number of these transient photocopies of the recipe of the mRNA and hand it to the chef, in this analogy, the ribosome, that will then make a certain number of proteins based on that. And the way this works is that a large part of how the chef determines how many dishes to cook, or how the ribosome determines how many proteins to make, is by the number of photocopies of that recipe that are available at that time. And it's very useful that the photocopies that the messenger RNA have a limited half-life, because that means once these mRNA, once a particular set of mRNA has perished, then the cell can make a new set of mRNAs and change the recipes, change the dishes that are being offered in the cell. So the very fact that mRNAs have a transient half-life is extremely important for dynamic changes in gene expression in this way. So I mentioned before also that our genes are split into exons and introns. And so the exons are like steps in a recipe, if you like, and a splicing process, which is when RNA polymerase transcribes a faithful copy of a gene, including the exons here depicted by these colorful boxes, but then including also the intronic sequences, which are then spliced out to make a mature mRNA or a readable recipe, if you like. So the splicing process makes a readable recipe, a mature mRNA. But what it can then also do is it can splicing can occur in alternative ways. So some splicing processes can skip an exon or retain an intron, various combinations of this sort of alternative splicing phenomenon. And the analogy that I come up with here is that alternative splicing is like making variant cakes. For example, you might have a cake recipe that includes an icing on the top, or you might have the same version of the cake, but plain without the chocolate on top. And so if you splice the recipe in different ways, you can either include the exon or chapter that specifies the making of that icing, or you can leave it out, and then the chef will make either a cake with or without icing. And that's how ribosomes interpret the genetic information provided to them in the form of mRNA. If they read whatever splicing has produced as an mRNA for them to turn into a protein. So I mentioned that we have only about 20 or 22,000 genes in our genome, and that is only about 2% of the genome sequence that has been sequenced around 2000 for the first time, and now many times over with many individuals around the world. So the rest of that DNA was initially thought to be some sort of evolutionary junk that had accumulated. But the reality is likely more complex than that, because for one thing, the more complex an organism becomes, there is a tendency for a higher proportion of junk DNA to be in their genome. Whereas the number of protein coding genes may remain relatively constant. So an alternative hypothesis that is gaining more and more support is that much of that non-coding DNA is not likely to be junk, but likely to have some form of important regulatory functions that helps with the production of the recipes in the right number and at the right time, if you like. So maybe an analogy that somehow works is to say, okay, a simple organism still has the same major parts. A simple plane has the same major parts as a very sophisticated plane, but the extent of regulatory control and so on that needs to be exerted to make that sophisticated plane perform as well as it does is really what differentiates the very rudimentary plane from a very sophisticated one. So regulation of gene expression becomes ever more important the more complex an organism becomes. And non-coding DNA is an important part of that regulatory network that allows that kind of complexity. So we can extend the central dogma of molecular biology that DNA makes messenger RNA, which is then translated into protein, by adding these non-coding DNA regions, and they're often transcribed into what we call non-coding RNAs. And so these non-coding RNAs are an important component of this regulatory network. They act both on other types of RNAs or back onto DNA and how it is expressed, or they can even regulate the function of proteins or be regulated by proteins. So you have this complex regulatory layer consisting of non-coding RNAs that helps cells and organisms to achieve complex the complexity that we have. So now I have a section on going back to messenger RNA and how they actually function in cells. So I mentioned before that genes encode or harbor the information, the genetic information of how to make proteins. And this was first deciphered in the middle of the 20th century onwards from there. And it led to the description of the genetic code, which basically says that the 4 letters of DNA in genes in DNA can be put together into triplets. And each triplet encodes for one of 20 amino acids, which are the building blocks of proteins. So the simple logical relationship then is that depending on what 3-letter code you see in a DNA sequence or in a messenger RNA sequence, which is a copy of the DNA sequence, is that each triplet codes for a particular amino acid. And because we have only 20 amino acids and 64 different combinations of triplets, the code is somewhat degenerate so that multiple triplets encode for the same amino acid. But there are also some codes that have special meanings. So the AUG codon not only encodes for methionine, but also signifies the start of what we call the reading frame. So a series of triplet words starts with an AUG. And then there are 3 codons that signify the end of a region that is to be translated. So when the ribosome encounters one of these 3 stop codons, it will end the synthesis of a protein and release. So here on the left, you can see a kind of schematic of how the ribosome goes through these different stages of first recognizing an mRNA and initiating translation, then elongating or engaging with the elongation phase of translation, which means that now a full ribosome is reading through one triplet at a time the sequence in the messenger RNA and uses the tRNA adapter molecule, bringing in amino acids to then add on to a growing polypeptide chain. And in the end, at a stop codon, the ribosome is released and also the completed protein is released. So a messenger RNA vaccine, obviously, then is an engineered messenger RNA molecule that is introduced into cells and that engages with the normal cellular process of mRNA translation. And for the duration during which this messenger RNA that has been introduced into the cell lasts, the cell will now make this introduced protein. And then once this messenger RNA is degraded after several hours or maybe a day or two, then the production of that foreign protein will cease. In terms of vaccination, that is, of course, what you want. You don't want a permanent expression of that foreign spike protein in your body. You just want it to be made for a brief period of time for the immune system to develop a response. But there are some other problems with protein synthesis that this diagram here doesn't completely explain. And that is, if you think about a long sequence of 4 bases in multiple combinations, if you read the triplets in the right reading frame, as we call it, they make sense. But if you start one base further along, then suddenly every triplet has a different meaning. So what is very important is during the initiation phase of translation that the ribosome knows where to start reading the code. And in bacteria, this is particularly important because bacteria transcribe their genes typically in the form of polycistronic messenger RNA. So this is like a schematic of a long messenger RNA made by a bacterial cell as a copy of multiple genes in their genome. And so if you have multiple genetic codes for proteins on the same string of RNA, you need to really know where to start and stop for the first cistron and start and stop for the second cistron and so on. So how do bacteria do this? How do bacteria find the right start position for translation? This was a question in the early 70s that people were trying to address. And John Shine and Linda Algano here at ANU made a breakthrough discovery. They were looking at some very by hard work obtained short sequences of ribosomal RNA. They were making a comparison with the sequences upstream of the start codon of some bacterial messenger RNAs that had also been sequenced at that time. And they realized that there is a potential for base pairing between a ribosomal RNA segment near the 3' end of the small ribosomal subunit RNA and a sequence just upstream of the start position of bacterial cistrons. And so they proposed at first that this is how the ribosome finds the right start, that this base pairing between these two complementary short sequences would achieve that. And in letters and so on, they corresponded way back then when there was no internet with Joan Stites and Karen Jakes in the US. And Joan Stites then picked up on their suggestion that this is how it might work. And they provided the first experimentally proof of this hypothesis. And they also coined the phrase Shine-Dalgano sequence for this short sequence upstream of start codons in bacterial mRNA. So understanding how bacteria direct their ribosomes to the right start position on messenger RNA was critical not only to understand bacterial biology, but then also enable the development of any biotech process that uses genetically engineered bacteria to achieve something, notably, particularly the expression of recombinant protein in bacteria as drugs and for other purposes. So this was really a key discovery that spawned a whole industry, if you like, as we know it today. Now, in higher cells, in cells with a nucleus or eukaryotic cells such as ours, messenger RNA has to function somewhat differently. First of all, genes in eukaryotic DNA are typically transcribed as a monocistronic mRNA. So the mRNA only has one coding sequence encoding one protein. But secondly, the mRNA is also then exported from the nucleus where the DNA is packaged away, if you like, to the cytoplasm where the ribosomes are. And around the same time as Shine and Dalgano were working on this conundrum in bacteria, other people were working on this question in eukaryotic cells. And Jerry Adams and Susan Corey at the Walter and Eliza Hall Institute in Melbourne published this seminal paper in 1975 where they had found a strange inverted nucleoside at the 5' ends of mRNAs, which they call bizarre 5' termini. And they were able to resolve the chemical structure of this cap as it became known. In parallel, Yasuhiro Furuichi and colleagues at the Roche Institute in New Jersey came up with the same discovery and also published in the same year. So having a cap structure for a eukaryotic messenger RNA is key for its function. And this was in part realized due to discoveries here in Australia. But mRNA vaccine, as I said earlier, has to retain all these features of cellular mRNA. So to make a successful mRNA vaccine, the companies who produced this mRNA had to make sure that this messenger RNA did not just have the coding sequence for the spike protein, but it also had to have a cap at the 5' end. And it's also important for efficient translation and stability of an mRNA to have a poly-A tail at the 3' end. So but these vaccines were designed to have both of these features. So now I'm going to run very quickly through how the cap structure and the poly-A tail are important to produce effective translation of an mRNA. And I'll do that just by comparing a schematic of how an mRNA vaccine was designed and some alternative features that might be included here. And compare that to a schematic of a messenger RNA and the different types of regulatory processes and translation processes that occur here. And you can see here also how exciting it was for me personally that a research area that I'd been working in since the mid-90s suddenly became the underpinnings of such an important new development in vaccinology and so on. So basically, a messenger RNA vaccine is made to look like a cellular mRNA and introduced into cells to temporarily force the cell to produce a foreign protein. But what else could we do with messenger RNA beyond vaccines, perhaps even, right? So let me see what the time is looking like. I need to speed up a little bit. But just maybe to emphasize that Katalin Karikó and Drew Weissman received the Nobel Prize in 2023 for their key discoveries around how mRNA can be introduced into cells and be productive in cells. In addition to the cap structure and the poly-A tail, they realized that certain internal nucleoside modifications were quite important to add to the messenger RNA to suppress an innate immune response that cells would otherwise mount if you introduce an mRNA from the outside. An mRNA, this is a summary of how an mRNA really is designed and produced. So you have a design part, you have a production of a DNA template, which is then used for a transcription process to prepare and purify the messenger RNA. The messenger RNA is then entrapped into a lipid nanoparticle. And then, of course, there are multiple stages of pharmacological evaluation of safety and efficacy before it goes into clinical trials and ultimately ends up being a useful vaccine or other therapy. So what are other applications of this mRNA technology that could be conceived of? Well, first of all, you could conceive of multiple other infectious diseases that now might be battled with an mRNA vaccine as an additional technology for vaccine production. But there could also be other applications that are to do with treating genetic disease or even non-genetic disease. And so principally, this revolves around what protein you choose to be encoded in your messenger RNA. So for example, in the case of a genetic disease where a gene is faulty and therefore a protein that is important in particular cells in the body is missing, you could introduce an mRNA into the patient cells to lead to the production of the missing protein and therefore correct the genetic defect. Now, the reason why this is attractive is that it's a transient treatment, right? So you'd have to have repeated doses of the mRNA to maintain the level of that therapeutic protein in the patient cells, which in itself is it can also be a disadvantage in that you have to have repeated dosing, but the big advantage is that you don't modify permanently the patient and its genetic material, or his or her genetic material. You you apply a transient treatment, which you can also withdraw if it is showing side effects or if it's no longer necessary. So there's a range of common and rare diseases to which now there are developments ongoing to develop mRNA therapy for these diseases. And you can also conceive of diseases that are common but are not necessarily genetic diseases, and conceive of mRNA drugs that can provide therapeutic protein in many of these different contexts. So with the proof of mRNA technology in the form of vaccines having been so clear, and also the companies that have sold the mRNA vaccines having made so much money from it, and others invest into this technology based on its performance as well. There's now a lot of effort around the world, both in academia and in industry, to come up with a multitude of these mRNA applications, and we can look forward to seeing many of these applications ultimately emerging in the clinic in the next decade or so, I think. So with that, I think I'll have to cut this last bonus section short so we have some time for questions and answers. But I just wanted to give you a flavor of how even a small country in terms of population as Australia has, how we have made quite some notable contributions to RNA science over the decades. So we have colleagues who have made early discoveries around RNA viruses, around RNA being able to catalyze reactions and be an enzyme, how alternative splicing is involved in HIV biology, how gene-mediated, double-stranded RNA-mediated gene silencing, which is now called RNAi, was discovered very early on in plants here in Canberra. We had colleagues like John Mattick who were very early on proposing that the idea of junk DNA is likely not correct. We have some antisense therapies that use splicing to correct a genetic defect being developed in Perth in Western Australia and going all the way into the clinic. We have interesting discovery science in RNA around novel structures in the nucleus that are involved in spatiotemporal control of gene expression. Just recently, the Nobel Prizes were announced, and Victor Ambros and Gary Ruvkun won the Nobel Prize in Medicine for discovery of regulatory microRNAs. Initially, they were seeing them in the worm, but here you have some examples of Australian researchers making notable contributions to how microRNAs function and how they have important roles in key physiological processes. And Ross Hannon, my colleague here at ANU, when he was working at the Peter MacCallum Cancer Institute in Melbourne, was a key driver in developing a small molecule drug that inhibits the production of ribosomes, and that is now in the clinic to treat cancer. And as it was said at the beginning, when I was introduced, we have recently started a Center for RNA Innovation here at ANU, and we take the name of the Shine-Dalgarno sequence to name our center as an inspiration for what we want to achieve here, driving forward both discovery science in RNA, but also translation of our findings into medicines and other biotech applications. And if anyone is interested in doing postgraduate studies or working as a scientist in Australia, I would recommend having a look at ANU and in particular our Shine-Dalgarno Center. We have, for example, quite attractive PhD scholarships that we hand out twice a year. And the next deadline is early next year. And with that, I finish, and I'm happy to take questions.
Thank you very much, Professor Preiss, for that very interesting talk, that very interesting lecture. We don't have any questions as yet, so I'll encourage the audience to ask any questions they might have. I found myself taking notes, actually, so I do have a couple of questions that I hope are relevant, keeping in mind that I don't have a science background. But yes, I'll go ahead and get the conversation started if you don't mind. You mentioned a couple of areas in which mRNA can go beyond vaccines. Which of these ones do you think show the most promise? I read cardiovascular disease and that sort of thing.
Yeah, I think what is probably most advanced at the moment is its use in cancer immunotherapy. And there it's a little bit related still to vaccine, the vaccine application. But basically what you can do is you can transiently program cells of the immune system to react to features of tumor cells. So tumor cells have genetic abnormalities that you can identify by sequencing them. And this could either be the case for a class of tumors across a range of patients that have that type of tumor, or it could be highly individualized for individual patients. But anyway, basically what the mRNA would be used there is to program the immune cells to develop an immune response to the tumor cells, but leaving the rest of the body unharmed, let's say. That's something that is quite advanced in development, and I think this will probably be among the earliest additional applications that we'll see in the clinic. There are other applications that are to treat genetic disease. There, a big challenge is the targeted delivery of the mRNA to the cell type or tissue where the genetic defect manifests itself. And the default cell type where LNP packaged mRNA gets delivered to is the liver. So I think in terms of genetic disease, probably genetic metabolic disease that manifests in the liver is going to be a field where this is going to be successful most readily, because the delivery is not a big problem. But many other genetic diseases, while conceptually is the same approach, you have to really innovate the delivery of the mRNA so that the message gets to the right cells and not broadly distributed in the body.
Thanks for that. Very promising times indeed, I guess. And a bit, you mentioned some challenges on that. So to follow up, I took a note here on potentially any ethical challenges or problems faced by developing mRNA therapies. Everybody remembers there was a big thing happening during COVID times around these vaccines. So if you can talk a little bit about that.
Yeah, sure. I think one of the things that happened during the pandemic is that everything had to be done so fast. Okay, so then we ended up in a situation where the general public was treated with a new vaccine technology without any time really to run an engagement or an information campaign or some a familiarization of the general public with this technology and what had been done to make it safe. Right. And so I think a lot of the vaccine skepticism that was directed particularly against mRNA vaccines simply comes from the fact that there was no time to explain this properly.
Yeah.
Having said that, I hope with some of my slides that I had early in my presentation, I tried to show you how different types of vaccines aren't really fundamentally so different, right? It's all about achieving the same goal, but by slightly different, plugging the technology of the vaccine into different stages of the gene expression pathway in the cell. And to my mind anyway, mRNA is conceptually particularly attractive and potentially particularly safe because it's an informational molecule, but not like DNA that is stable and may stay with you long term. It is transient, right? So you inject a small dose of that. It is hanging around for a day or two, but then it's gone. So there's no permanent genetic modification of a patient occurring in this approach. And
It's
all
usually
it
the
does
case,
really
right?
is lead to the transient production of that foreign antigen that the immune system develops a response to. So it's really very flexible and intrinsically safe in that way.
Yeah,
That's
thanks
not
for
to
that.
say that mRNA vaccines have zero side effects. No medical treatment has absolutely no side effects. But if you take a hard look at the statistics, or even of side effects of mRNA vaccination, you'd have to understand that they have been exceedingly rare.
Mm.
Right. I mean, it is almost unprecedented how many people were treated with this novel drug, if you like, around the world. And then compared to the number of people that received the treatments, the number of people with serious side effects was very, very small. I mean, that doesn't diminish the seriousness of side effects that individuals suffered, perhaps. But this is just that is the nature of medicine. That nothing is like 100% foolproof.
Guaranteed. Absolutely. We did receive, thanks for that. We have now a couple of questions that just came through. So with a couple of minutes left, I'm going to read them to you if you don't mind. How exactly do you target these nanoparticles to particular cell types? Because I assume random expression in random cells might be dangerous.
Yeah, that is the key question, right? I don't think anyone has the comprehensive answer to that, but there are some developments that I think are quite promising. So one thing is to engineer the lipid molecules to be somewhat semi-selective in terms of the cell type where they fuse with the cell membrane. And so I think compared to what the vaccination in the pandemic was run on, there is potential there to innovate. But then you can also try and engineer something on the surface of the nanoparticles that will provide a more direct homing strategy. So you can, for example, decorate lipid nanoparticles with a protein moiety that will bind to a certain receptor that is going to be present only on a certain cell type in the body. For example, let's say cardiac cells. If you have a binding surface in the heart that you can engineer a protein for that will selectively bind to that, then if you decorate your lipid nanoparticle with such a functionality, then it will drive selective accumulation in the target tissue. So I think that those are some of the strategies that are being developed to achieve that. But you can also work on the mRNA itself. So an mRNA is true if a protein is made in the wrong cell type, that could potentially be harmful. So, but if the mRNA requires a certain environment to be translated, then a certain level of introduction of the mRNA into a wrong cell milieu wouldn't matter as long as it doesn't get translated there. So you can engineer the mRNA to be preferentially translated only in the target cell type, or you can preferentially lead to the degradation of the mRNA in the wrong cell type, so that even if the LNP delivery isn't particularly selective, then the expression of the mRNA can be more selective to a target cell type.
Thanks for that. The next question here is, what is the expected duration of immunity from an mRNA vaccine, and how does it compare to other types of vaccines?
Well, I think that's a question that I can't honestly answer from a perspective of a lot of knowledge. I'm not a vaccine expert. I'm an RNA biologist. So I learned a lot about vaccine technology through my interest in the pandemic and how we were dealing with it. But broadly speaking, the immune response triggered by the mRNA vaccines is of a similar nature than that to other vaccines. So no vaccination necessarily lasts permanently and needs a refresher. I think the bigger problem we have with COVID vaccines and with many other vaccines for infectious disease, for example, flu, is that the pathogen mutates and changes. And so what is potentially still a viable immune response that your body has from the last vaccination is no longer effective because the virus has changed. That's why you need to have a vaccination with a variant vaccine. With flu vaccines, we're very familiar with that. Every season, you need to get a new flu jab to be vaccinated against the newly involved flu virus. And we're looking at a situation now with COVID that is similar. You know, we hear about new variants of COVID emerging and becoming dominant in the infectious arena, if you like. And so particularly vulnerable populations among us have to be vaccinated with vaccines that are going to present to the immune system a spike protein that represents that variant virus.
Thanks for that. The last question we have here is also vaccine related. I'm going to read it anyway.
Yeah.
Have we mastered the development of mRNA vaccine? If we are to choose mRNA vaccines for the next pervasive infectious disease in the near future, is the technology amenable to adapt that quickly?
Yeah, I think certainly the speed with which you can design and produce mRNA vaccines is unparalleled. So speed is a very big plus for mRNA vaccines and flexibility. But having said that, producing mRNA vaccines at the moment is more expensive than some of the more conventional mRNA vaccine technologies. So if money is critical, for example, treating people in the developing world where money is scarce, then mRNA has a disadvantage there still. Although having said that, I know that there are now mRNA vaccine production facilities even in Africa, in places and so on. So I think over time the cost will come down for mRNA vaccines. But the third thing is still that different types of pathogens may require different types of vaccine technology for particularly effective vaccines as well. So this, I don't think that the inactivated virus approach, the very traditional vaccine approach, is dead yet, because that is still a way in which you can present the immune system with the pathogen in the most holistic way, if you like.
Thanks for that. Okay, we have gone slightly over time. Professor Preiss, thank you so much for your time. Thanks so much to the audience today, and for those of you who have submitted to all of you who have connected, who have come online, and those of you who have submitted your questions, of course. Thank you very much.
All right, thank you.
Bye-bye.
Bye-bye, everyone.